IB Bio Cell Structure and Function Part 4 will focus on eukaryotic cells, cells with a nuclear membrane that houses the DNA, and other internal compartmentalization. The essential idea is that eukaryotes have a much more complex cell structure than prokaryotes. Here is the outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused here, and this is the first of two movies on eukaryotic cell structure. Scientists have grouped all of the world's organisms into three domains, seen here in purple: the archaeans, the eubacteria, and the eukarya. The archaeans and eubacteria are prokaryotic. Their cells lack compartmentalization. They lack a nuclear envelope around the DNA. The eukarya domain includes all organisms whose cells have a nucleus, including protists, fungi, plants, and animals. Remember that viruses are non-living biological particles. Here's a diagram displaying the three domains: the archaeans, eubacteria, and eukarya. The common ancestor is here. Archaeans are prokaryotic cells that tend to live in extreme environments, hot or salty, sulfurous. Archaeans have certain characteristics that suggest they are more closely related to eukarya than eubacteria are to eukarya. The characteristics of eukaryotic cells are that they are larger than prokaryotic cells, 10 to 1,000 micrometers. There's a nuclear membrane around the DNA, internal organelles such as the chloroplast and mitochondrion, numerous linear chromosomes composed of DNA and histone protein. The DNA has non-coding sections. The DNA is wrapped around protein, histone protein. The ribosomes are larger than prokaryotic ribosomes at 80S. Here's the first IB syllabus statement that provides the focus of this movie. Draw a diagram of the ultrastructure of a liver cell. Include the following structures: free 80S ribosomes, rough endoplasmic reticulum, rough ER, lysosome, Golgi apparatus, mitochondrion, nucleus. State the function of each structure. The specification of liver cells simply provides some focus on what type of eukaryotic animal cell to examine. Here is the first of many images of a cell. Let me briefly cover the structures that will be important for you to get to know. Any structures I don't mention are structures for which you are not held accountable by the IB syllabus. We have the plasma membrane. It's the boundary that maintains the integrity of the cell and serves to regulate what enters and exits a cell. We have the nucleus, which houses the eukaryotic DNA, many linear strands of DNA, all of it associated with histone protein. We have the cytoplasm, a watery suspension in which chemical reactions occur. We have the endoplasmic reticulum, aka ER, its membrane studded with ribosomes. Ribosomes serve as the site of protein synthesis. Notice that some of the ribosomes are associated with the ER, while some are free in the cytoplasm. I'll speak more about this later. The Golgi, like the ER, is composed of membrane and serves to modify and transport protein. Vesicles pinch off from the Golgi, transporting protein to the plasma membrane. Lysosomes are compartmentalized structures holding enzymes that digest food taken in by endocytosis, or the lysosomes have enzymes that digest old organelles, old worn-out mitochondria. Mitochondria is the site of energy transformation, aerobic cell respiration. Here is your second diagram of an animal cell. You can see the plasma membrane, you can see the nucleus, and there are chromosomes within the nucleus. Remember, eukaryotic DNA is composed of many linear strands associated with histone protein. Notice the endoplasmic reticulum with some ribosomes. As well, there are ribosomes free in the cytoplasm. Ribosomes synthesize protein. The Golgi will receive vesicles that have pinched off the ER within which there are protein molecules. The Golgi vesicles will pinch off from the Golgi and take protein to the plasma membrane to be released from the cell. The mitochondrion transforms energy through aerobic cell respiration. Here is the third diagram of an animal cell. I want you to study the structures in the diagram. This image nicely shows the large pores in the nuclear envelope. I'll come back to those later. While the endoplasmic reticulum seen here and the Golgi seen here are both composed of membrane and both compartmentalize the cell, I want you to work to differentiate their structure. The ER is typically located closer to the nucleus and is likely to be associated with ribosomes. The Golgi is never studied with ribosome. Here is your first look at a plant cell. Let's focus on the differences relative to an animal cell. The regular, somewhat geometric shape is provided by the presence of a cell wall. This serves to protect the cell. It's composed of cellulose. Animal cells never have a cell wall. Just under the wall is the plasma membrane in blue. All cells everywhere have a cell membrane. The plant cell also has a large central vacuole that serves to maintain water balance. Chloroplasts function in photosynthesis and are not found in animal cells. All of the other structures should be familiar to you. This plant cell diagram is very good, nicely distinguishing the following features: cell wall from cell membrane, ribosomes on the ER from free ribosomes, mitochondria from chloroplasts. The internal compartmentalization of the eukaryotic cell is well displayed with this image. As a plant cell, whose chloroplasts are responsible for photosynthesis, the presence of a starch storage vacuole might not surprise you. The sugar formed through photosynthesis is stored as starch. Remember, ribosomes are large enzymes that synthesize protein. Here are the functions of the nucleus of a cell, the cell membrane, and the cytoplasm. You should study this slide on your own. Here are the functions of all the structures I've mentioned so far in this introduction to eukaryotic cells. Endoplasmic reticulum with vesicles that pinch off, modify, and transport protein. Ribosomes construct protein. Golgi apparatus with vesicles that pinch off, modify, and transport protein. The lysosome digests worn-out organelles, digests food within food vacuoles, and vacuoles serve to store materials such as starch, water, and ions. Can you identify and state the function of A? This is the plasma membrane. It serves to regulate what enters and exits a cell. B is the nucleus. It's housing many linear strands of DNA molecules. C is the nucleolus. This is the region of the nucleus where ribosomes are assembled. D and E together is the endoplasmic reticulum and ribosomes, together known as rough ER. Protein is synthesized by the ribosomes and then modified and transported by the ER. F is smooth ER. Let's leave the function of smooth ER aside for now. G is Golgi. Golgi serves to modify and transport proteins. You can see the vesicles carrying protein toward the plasma membrane. Mitochondrion H is for energy transformation, aerobic cell respiration. You can study this slide on your own for the identification of structures and work to be able to state basic functions of each. Here's a nice diagram that distinguishes a plant cell on the left and an animal cell on the right. In this image, the cell wall and the chloroplast are the only structures not common to both cell types. Here's the relevant IB syllabus statement. State with detail at least 5 differences between a typical plant and animal cell. You can review your understanding of plant cells with this slide. B, C, and D, all of which constitute A, represent components of the nucleus, the nucleoplasm, the nucleolus, and the nuclear envelope. E is endoplasmic reticulum, while F identifies ribosomes on the ER. G is the large central vacuole, typical of plant cells. H identifies the membrane of the central vacuole called the tonoplast. I identifies a chloroplast. J is the cell wall, K is the plasma membrane, L is a mitochondrion, and M is a Golgi vesicle pinching off of N, the Golgi. More review. Turn the movie off now or follow along with me. A, cell wall. B, cell membrane, plasma membrane. C, cytoplasm. D is a mitochondrion. E is a chloroplast. F, starch storage vacuole. G is the cell membrane of the adjacent cell. H is the cell nucleus. And I is the central vacuole with a membrane, the tonoplast, that's large and typical of plant cells. This is a micrograph of cell wall, cellulose, cell wall of a plant. You can see the fibrous nature of the cellulose. The cell wall serves to provide a plant cell with structural integrity, serving to assist in holding a plant upright. In terms of cell structures, it's important to remind you of cilia, seen here on the paramecium, and a flagella, seen here on this sperm cell. Both of these structures serve to provide motion. Here's a comparison of animal and plant cells. Hopefully, much of what you find here would be review, but there are some differences that I have not emphasized through the course of this movie. Animal cells have many small vacuoles, no central vacuole, while plant cells have a large central vacuole. Animal cells have many lysosomes. As cells that would ingest organic molecules, they need lysosomes to digest those organic molecules. Plant cells, on the other hand, as autotrophs which synthesize organic molecules through photosynthesis, would have few, if any, lysosomes. The carbohydrate storage molecule in animal cells is glycogen. The carbohydrate storage molecule in plant cells is starch. And lastly, animal cells have intercellular connections, known as gap junctions, while the intercellular connections in plant cells are known as plasmodesmata. Many of the structures stated here will appear in discussions later on in this course that would include gap junctions and plasmodesmata. So take in the high detail here if you can, on the hopes that it will help strengthen what you do later on in IB biology. And that brings us to the end of IB Bio Cell Biology Part 4.